Hello and welcome to Nikolay's genetics lessons, and today's problem is: in an average human population, approximately 8% of the males are colorblind. Remember that this is an X-linked recessive trait. Assuming Hardy-Weinberg proportions, answer the following. Question A: what proportion of females would you expect to be colorblind? And now let's use a Hardy-Weinberg formula in order to solve this problem. So frequencies of the three genotypes that can be made by two alleles would equal to p squared plus 2pq plus q squared, and this would equal to 1, or the three genotypes would represent 100% that can be made by two alleles. So in this formula, p squared stands for the homozygous dominant normal genotype. So females would belong to this genotype, would have two normal X chromosomes. 2pq would stand for the heterozygous genotype, so also p would stand for the normal allele or X chromosome, and q would stand for the defective X chromosome, or in other words, for X chromosome with defective allele on it. And q squared would stand for the chromosome that is going to be defective, and no other chromosome, because in this variant we would see affected males, and males has only one X chromosome. Another chromosome would be, sex chromosome would be Y chromosome. So this is a tricky problem. If we are told that 8% of the males would be affected with this genetic disorder, that give us, so imagine this is like cloud, and this is gene pool. In this gene pool we would have normal X chromosomes, and we also may find here X chromosome that is going to be defective. And this defective X chromosome, when would be inherited by male, would manifest itself as genetic disorder. So usually, in order to find frequency of the q, we have to take a square root of q. But in our example, we don't have to take square root, because male is going to be hemizygous, and if we know that male, 8% of the males would be affected with this genetic disorder, that gives us information that 8% of the X chromosomes in the gene pool would carry this defective allele. So now we can solve for females that would have two X chromosomes. None of these genotypes would be male genotype, because males do not have two X chromosomes. So according to our problem, we have to find what proportion of females would you expect to be colorblind. So let's add another defective X chromosome here, and this would represent defective phenotype of the female, when both X chromosomes would be defective, and probability of such an event would be, so 8%. We know that this defective X chromosome represent 8% of the gene pool. In other words, we can say this is 0.08. So probability that one of the X chromosomes would be defective would be 0.08, and probability that the second X chromosome also would be defective also would be 0.08. So now we have to multiply all these probabilities, these two probabilities, and this would give us a probability of such an event as 0.0064. Or if you need an answer in percentage form, you have to move this decimal point two places to the right, and the answer would be 0.64%. So a little bit more than 0.5% of our population, human population, or 0.64% of the females would be colorblind. So when you are told that females do not have color blindness, this is not true. But males, of course, would have or manifest this genetic disorder to much higher extent, as you see 8% here and 0.5% here, because males are hemizygous, and for male probability would be 8% because 8 X chromosomes out of 100 would be defective. So, next question, what proportion of females would you expect to be carriers of the recessive allele or would be heterozygous? So, according to our formula, we have alleles P and Q. Allele P stands for the normal phenotype, normal allele on the X chromosome, and recessive allele Q means a defective X chromosome. So allele P plus allele Q should give us 100%, because as you see, 3 genotypes here are made by only 2 alleles. So we have only 2 types, as you see, green and red. So if we know that frequency of the Q allele is 0.08, this gives us information that frequency of the P allele, which is normal, would be 0.92. So the number of heterozygous females or females who is going to be carriers, according to our problem, according to our formula, would equal to 2pq, or 2 multiplied by number p, which is 0.92, and multiplied by number q, which is 0.08, and we are going to get 0.1472. If we need an answer in percentage form, we have to move this decimal number 2 places to the right, and the answer would be 14.72%. Now, when we know frequency of the homozygous recessive genotype, which is equal to 0.0064, and we know also frequency of the heterozygous genotype, which is 0.1472, we can find frequency of the homozygous dominant genotype. We just have to add these two numbers and subtract from 1, which would give us frequency of the homozygous dominant genotype as 0.8464. So when we add these 3 frequencies, we should get 1 or 100%. So these 2 genotypes would make normal phenotype, because this is X-linked recessive genetic disorder. So females who is going to be heterozygous would be of the normal phenotype, and only those females who is going to be homozygous recessive would be affected. So we would have here 2 phenotypes. And the last comment would be, you have to remember that females also may be affected with X-linked recessive genetic disorders, but this is going to be a product of occurrence of this genetic disorder in males. For example, if we would see 8% in males, in females we have to multiply this number by itself. If, for example, hemophilia would have occurrence in males like 1 in 1000, in females this is going to be 1000 multiplied by 1000. So in females this would occur 1 in a million. So as you see, this is tremendous difference explained by the fact that males have only one X chromosome and females has 2 X chromosomes. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates and see you in the next video. Goodbye.